The title of our talk today is The Persistence of Transplanted Explant-derived Human Cardiac Stem Cells Contributes to the Therapeutic Regeneration of Injured Myocardium. This was a study performed in my lab by students, and most responsible for this study is Elizabeth Richardson. As you can see, heart failure is a malignant disease, and this is the target of our therapy. Here's the projected lifespan of a healthy 65-year-old patient. When this patient is first diagnosed with heart failure, their survival plummets. Only one hospitalization for heart failure, such that 50% are dead at 5 years. The hopes for regenerative therapy is that this can restore our patients back from this red line to the healthy 65-year-old expected mortality. What are explant-derived cardiac stem cells? These are stem cells which are actually derived from human tissue. What we do is we take a piece of human tissue on a biopsy, or we take a piece of specimen obtained at the time of surgery and place it in culture. This is the piece of tissue here. Over time, cells grow out from this tissue and cover the cell culture ware. We harvest these cells, and these are explant-derived cardiac stem cells. Transplantation of these cells are able to repair injured myocardium. They rescue damaged tissue, they encourage endogenous regeneration, and grow into new working heart tissue. However, only 1 to 2% of transplanted cells persist out to 4 weeks after injection. Therefore, it's unclear what the meaning of persistence of the cells is to these therapeutic endpoints. Therefore, our study question was, is it important for cardiac stem cells to survive and persist after transplantation? We used a novel technique to perform on-demand ablation of transplanted human cardiac stem cells to dissect the contribution of engrafted cells to myocardial repair. As you can see here, Diptheria toxin is able to kill human cells and tissue, but not rodents. Incorporation of a single molecule of active Diptheria toxin causes protein arrest and cell death. Mice and rats, however, lack the receptor for Diptheria toxin, therefore they're resistant. Here you can see human cardiac stem cells that express the Diptheria toxin outlined in green. The blue is the nucleus of the cells. The bottom graph, we took cells from mice and cells from humans. We exposed the mouse cells to Diptheria toxin, and there was no cell death. However, when we exposed the human cells to Diptheria toxin, there was 11-fold cell death over 24 hours. This proves that Diptheria toxin is capable of killing the human cells transplanted into mice. Therefore, what we did was we used immunodeficient mice that enable us to transplant in human cells. We gave them a heart attack during a surgical procedure, and then 1 week later we transplanted in human cardiac stem cells, or simple saline. We performed echocardiograms 1 week later, and then at that 1 week point, I injected these mice with either saline or Diptheria toxin for 5 days. And then we performed a series of echocardiograms to determine how well their heart functioned. Here you can see the cardiac ejection fraction, or the ability of the hearts to squeeze. 1 week after the heart attack, all mice had an ejection fraction of around 30%, normal being 50 and greater. So this shows that we were able to injure a significant amount of heart tissue. When the animals were injected with saline instead of stem cells, the heart function declined over time as they remodeled. When the animals were injected with stem cells but just saline, so no Diptheria toxin, the animals improved over time, as we've shown in previous studies. When we ablated the stem cells by injecting Diptheria toxin 1 week later, there's a marked decrease in the performance of these cells. Therefore, based on these findings, we conclude that this is the first use of Diptheria toxin to eliminate human cells after transplantation. We also conclude that elimination of the cells 1 week after transplant reduced cardiac stem cell mediated repair. The mechanisms behind this are likely persistent secretion of healing hormones and/or cell replacement into new working tissue that contribute to the maintenance of therapeutic regeneration. Thank you very much.